Hello, everyone. I am Briana de Miranda, and I am from the University of Alabama at Birmingham. And today I will be talking about what's new in environmental toxins. Before I start talking about the environment and Parkinson's disease, I thought I would first mention what is true for all disease, which is that all health is defined by our genetics combined with our environmental exposures. And this is what drives all disease phenotype, whether this is cancer or heart disease or even neurodegeneration. Now, this is particularly true for Parkinson's disease because of the high number of environmental contaminants that are linked to Parkinson's disease risk. And we can visualize this if we imagine Parkinson's disease incidence along a bell curve spectrum, where at one end of the spectrum, we have inherited genetic mutations that lead to familial Parkinson's disease. And at the other end of the spectrum, we have known environmental or chemical exposures that have led to the development of PD. And in the middle of this bell curve is where we see the majority of Parkinson's disease. So about 85% of individuals with PD fall within here, where underlying genetic susceptibility combined with a lifetime of environmental exposures leads to the development of idiopathic PD. And so in thinking about what environmental toxicants are associated with increased risk for PD, or this environmental part of the equation here, we can group this broadly into two categories: pesticides or industrial byproducts. It's worth mentioning that there are other environmental elements that can increase risk for PD. So for example, certain viruses are associated with elevated risk for PD. And then of course, diet and then pharmaceutical or recreational drugs can also play a role in whether an individual develops the disease. However, for the purpose of my talk today, I'll be talking about these two categories here. So pesticides have historically been the first environmental contaminants that are linked to increased risk for Parkinson's disease. And there are a number of pesticides that have been associated with elevated PD risk, and I'm not showing all of them here, but I would draw your attention to this compound here, paraquat. Paraquat represents a risk factor for PD predominantly in the United States, actually, because paraquat usage in the US is increasing per year, even as it is banned by other countries. So for example, the entire European Union no longer allows paraquat to be used. However, as certain crops become resistant to other pesticides, paraquat usage in the United States is growing. Paraquat is associated with increased risk for Parkinson's disease. And so we worry about paraquat exposure for individuals who live or work in agricultural communities, as well as pesticide applicators. In thinking about what's new in environmental toxins, I won't focus any more on pesticides simply because we've studied pesticides more exhaustively than we have other compounds that we think increase risk for Parkinson's. So while pesticides still represent a risk factor, I'll focus today on industrial byproducts, specifically industrial products like organic solvents, which elevate risk for Parkinson's disease, but have not been very well studied in the risk for PD. So for example, trichloroethylene is a very commonly used organic solvent that is a degreasing compound. And then perchloroethylene, or PCE, is the most commonly used solvent in the dry cleaning industry. Toluene is also a heavily used solvent, and all of these we think increase risk for PD. PCBs have historically been used, and these increase risk for Parkinson's disease. And actually, even as a number of these compounds are phased out of usage, so you might know that PCBs are no longer used in different industrial applications, you might notice that a lot of these compounds are chlorinated, and the number of chloride ions a molecule has, the harder it is to break down in the environment. And so even if these are not used in industry today, they may still represent environmental contamination and risk factors for us who live or work near where these are either stored or applied. And then metals actually represent a very historical as well as current risk factor for Parkinson's. So for example, lead, copper are some of the most heavily emitted compounds into the environment. And then manganese was traditionally or historically associated with a type of Parkinsonism called manganism, which we saw in welders who breathed in fumes that contained manganese. Fortunately, that has been reduced significantly by proper PPE. However, manganese still represents an environmental risk factor because manganese is a component of air pollution and can also be found as a drinking water contaminant. And so manganese represents a risk factor for individuals who are exposed to that today. And in thinking about certain populations that might be more or less exposed to these, we can use an example of people who serve in the military as an example of a population that has increased risk for Parkinson's based off of exposure to one or more of these compounds simultaneously. This, combined with other risk factors, for example, head trauma, has led to individuals who either currently or formerly served in the military being at elevated risk for Parkinson's disease. So as I mentioned, today I'm going to focus on this group of compounds here, the chlorinated solvents. Chlorinated solvents are a type of halogenated solvent, and there are 46 halogenated solvents in production today. These are an organic compound with a chloride, fluoride, bromide, or iodide compound on them, and they are really heavily used throughout the world. They are responsible for degreasing operations as well as used in chemical feedstocks. And a number of them have been associated with Parkinson's disease risk, as well as a number of them are inferred in Parkinson's disease risk, but we just don't have the data on them yet. And I'm most interested in this compound here, TCE. And there are many reasons for this. One reason is because TCE is one of the most widely used industrial solvents throughout the world. And even as TCE use is decreasing in the West, it is increasing in Asia. So it's still growing globally by about 2 to 4% annually. Another reason why I'm interested in studying TCE is because it represents a seminal compound in these different types of halogenated solvents. And so by studying TCE, we can infer the toxicity of numerous structurally similar solvents without having to study each of them individually. So how might you be exposed to TCE? TCE is emitted predominantly through the air of factories or industrial sites where individuals live near or work in. And so you might be exposed to TCE if you inhale it directly. Another way you could be exposed to TCE is when it is irresponsibly dumped into the ground. It gets into the soil as well as the groundwater, and when it hits groundwater aquifers, it travels in these non-aqueous plumes and gets underneath residential and commercial buildings, where it can then off-gas into the airspace of these buildings. So TCE is a volatile organic compound, or VOC. So even if it exists as a liquid in groundwater, it can volatilize into the airspace of a building. And so you might be secondarily exposed to TCE if it is within the airspace of where you work or live. You may also be exposed to TCE if it is within your drinking water. And actually, TCE is the most commonly sampled organic contaminant within drinking water. And this is true for the United States, as well as the United Kingdom and many other countries throughout the world. So TCE represents an environmental risk factor for exposure, as well as a risk factor for Parkinson's disease. And my lab wanted to understand a little better about how TCE causes the type of brain pathology that might lead to PD. And so what we did was we developed an animal model to study TCE, where we treated adult male Lewis rats with a daily oral gavage exposure of 200 milligrams per kilogram of TCE. We did this over a chronic time period for approximately 6 weeks in total. And then at the end of this 6 weeks, we measured open field behavior as well as brain pathology. And what we found was that animals that were treated with TCE had a significant reduction in spontaneous locomotor activity. And this was true for some, but not all of these open field parameters. And so this indicated to us that these animals had a pretty mild behavioral phenotype following TCE exposure. And because of this, we thought these animals may not be truly Parkinsonian because they had sort of a mild phenotype. And also they did not appear overtly toxic. They didn't have any signs of reduced motor capacity except for when we measured them on this sensitive assay. So what we found when we looked at their brain was very interesting. They had a significant reduction of dopaminergic neurons in the substantia nigra, as well as neuron projections into the striatum. And this indicates that they had a significant loss of nigrostriatal dopaminergic neurons. Interestingly, these rats did not show a loss of dopaminergic neurons in the VTA, which is true for idiopathic Parkinson's disease as well. So this does look like a true Parkinsonian lesion caused by systemic exposure to TCE. We were also interested in what was happening to these neurons before they degenerated, or what was happening in the neurons that survived here in the substantia nigra. So we looked at pathology of these dopaminergic neurons in TCE-treated rats. And one pathway we looked at was the endolysosomal pathway, the pathway that's responsible for degrading protein in the cell. And what we found was that Rab5 positive early endosomes, seen in red in the first panel here, were significantly accumulated in rats that were treated with TCE. And so this indicates to us that the early endosomes were not being properly matured into late endosomes and then lysosomes. And in fact, we also saw a significant reduction in lysosomes and late endosomes, which you can see here in lamp 1, which is in yellow. And these were significantly reduced in TCE-treated rats. We also found that the ubiquitin-like protein P62, which tags cargo destined for the lysosome, was significantly elevated in the dopaminergic neurons of TCE-treated rats. And taken together, this suggests that the endolysosomal pathway responsible for a type of protein degradation is dysfunctional in rats that were treated with TCE. It was not surprising then that we saw an increase in protein accumulation within these dopaminergic neurons, particularly alpha-synuclein. So for example, phosphorylated alpha-synuclein as well as total alpha-synuclein were significantly elevated in the dopaminergic neurons of TCE-treated rats. Next, we wanted to look at this genetic component of the gene-environment interaction. And for this, we turned to the protein LRRK2. Some of you may be familiar with LRRK2. This is probably because mutations in LRRK2 are the most commonly inherited mutations associated with familial Parkinson's disease. And mutations in LRRK2 result in a toxic gain of function of the kinase activity of LRRK2. This leads to an increase in phosphorylation of LRRK2's cellular substrates, most of which are Rab GTPases or RABs. And this leads to a number of cellular pathologies, including issues with the endolysosomal system. We can measure elevated LRRK2 kinase activity by measuring this phosphorylation that the kinase domain of LRRK2 induces. So for example, autophosphorylation at 1292 of serine 1292 on LRRK2 indicates increased LRRK2 kinase activity, as well as serine 935, which interacts with LRRK2's binding partners. We can also measure LRRK2 kinase activity by measuring phosphorylation of its Rab GTPase targets, such as Rab10. And so this is what we did in the brain of TCE-treated rats, where we took animals that had been treated with TCE for 3 weeks and looked for elevated LRRK2 kinase activity in their brain. At 3 weeks, we did not see any dopaminergic neurodegeneration that we saw at 6 weeks, so we knew that this was a pre-degenerative time point. And what we found was significantly elevated LRRK2 kinase activity in the striatum as well as substantia nigra of these TCE-treated rats. So serine 935 as well as serine 1292 and phosphorylated Rab10 were all significantly elevated in the striatum, and then serine 1292 is significantly elevated in the substantia nigra. So together, these data tell us that LRRK2 kinase activity was elevated in the brain region that degenerated in the TCE-treated rats prior to dopaminergic neuron cell loss. So this suggests that TCE significantly elevated LRRK2 kinase activity in the brain, even as we gave this as a systemic exposure. And interestingly, we are not the only ones who have found this. So for example, manganese-treated macrophages also showed significantly elevated LRRK2 kinase activity, as well as dopaminergic neurons in the brain of rotenone-treated rats. And interestingly, this study looked at phospho-Rab10 in rotenone-treated rats and found that when the small molecule LRRK2 kinase inhibitor, PF360, was co-treated with rotenone, there was a significant reduction of this elevated LRRK2 kinase activity. And this suggests that LRRK2 kinase activity that has been elevated by environmental exposures could be pharmaceutically modifiable by a LRRK2 kinase inhibitor. So taken together, what we've concluded from this is that wild-type LRRK2 activation could be a common feature of Parkinson's disease-associated environmental toxicants, and therefore increased risk for PD. This becomes particularly important for individuals who have a LRRK2 mutation, as already high levels of LRRK2 kinase activity combined with environmental exposures that further modify LRRK2's kinase activation status could lead to a Parkinson's disease phenotype through this gene-environment interaction. So how can we influence this phenotype, and how can we reduce Parkinson's disease risk with environmental exposure? Well, one way would be to use a targeted treatment of a LRRK2 kinase inhibitor, such as those that are currently in clinical trials, to reduce LRRK2 kinase activity as a result of these environmental exposures. This might be useful as a short-term treatment if we know that an environmental exposure has occurred and that LRRK2 kinase activity has subsequently been elevated. Another potential way we can inhibit LRRK2 kinase activity might be to actually try to inhibit LRRK2 kinase activity before it starts. So for example, all of the compounds I showed you today, TCE, manganese, and rotenone, all cause oxidative stress within the cell. And so by treating with a brain-penetrant antioxidant after environmental exposure, we might be able to intervene prior to LRRK2 kinase activity elevation and therefore subsequently reduce Parkinson's disease risk from these environmental exposures. And this would be useful in individuals who are in the prodromal stage of PD. So as we get better at understanding when Parkinson's disease develops, and if we can track these environmental exposures better, we might be able to intervene in time to prevent that PD phenotype from occurring. Another way we can influence disease phenotype is by influencing policy. And a number of policies need to be enacted in order to reduce environmental contaminant exposure. In the United States right now, we are trying to ban neurotoxic compounds that are associated with elevated risk for PD. So for example, paraquat and TCE. Some states have banned TCE, but certainly the minority of states throughout the US. And in other countries, this is true as well. We need to work towards banning TCE and other neurotoxic compounds like paraquat so that we can reduce exposure and try to get at that environmental side of the equation a little bit better. We also need to be cognizant of who is most at risk for exposure to these compounds. So people who live in underprivileged areas of the world and of probably your home country are at the highest burden for exposure to environmental contaminants. And so this creates a disease bias risk for individuals who have low socioeconomic status. And this is something that we need to enact policy in order to prevent, because people who live in these areas cannot themselves clean up the air and the water in which they live. And then lastly, I think we need better analyses for what we are exposed to on a daily basis. So monitoring the air and the water in your home as well as where you work could be really important. And I think that the government really needs to intervene in this because this is really too expensive for the individual to take on themselves. And so with all of these things combined, we can start to get at that environment side of the equation a little better in order to influence PD phenotype. On the research side, one way we can influence the environment is to incorporate more environmental exposures into PD research. And so not just using these compounds as neurotoxins that increase dopaminergic neurodegeneration, but really trying to understand how environmental exposure can elevate risk for PD. And then lastly, we need to kind of push the envelope a little bit and start looking at multiple exposures to get at that environment-environment interaction, as well as genetic susceptibility to look at gene-environment interaction so that we can kind of get at what would be a true exposure in human populations in order to better model this and predict Parkinson's disease risk. So as I conclude today, I'll leave you with a couple of resources. If you're within the United States, you can go to the EPA.gov to learn more about the compounds I talked about today, as well as the ATSDR. This is a website that is controlled by the CDC, and they have really good information on different toxic substances that can lead to elevated risk for disease, as well as where these toxic substances are in the areas that you live. If you're outside of the United States, the Pollutant Release and Transfer Register is a website that has links to different government websites from respective countries, and this can have information for you about what pollutants are released in your area. So as I conclude today, I just want to thank you for your time and listening to me. And I also want to thank the organizers of this session for an opportunity to talk today about this important subject. And I look forward to talking to you more about this in just a few moments.